All right. Hello, everyone. We will be starting right now. Thank you so much for coming to this Invasive Crayfish Collaborative webinar, the third of our series. My name is Natalia Shklaruk, and if you don't know me, I'm with the Illinois Indiana Sea Grant and am the main facilitator of the Invasive Crayfish Collaborative, which is a program that brings together a variety of experts and stakeholders to address the threat of invasive crayfish. Now, before we begin, I wanted to share a list of different links that you can follow if you are interested in learning more. Please check out our main website, invasivecrayfish.org, which acts as a one-stop shop for invasive crayfish information and is always being updated. And if you're interested in joining the ICC membership, you can follow the join link to find our subscription form. And if you're also interested in registering for our future webinars or viewing past webinars, you can follow the last two links. And then finally, you can email me at shklaruk@illinois.edu if you have any questions or comments. And I believe we are adding these links to the chat so you can easily access them yourself. So one of the goals of the ICC is to make new information regarding invasive crayfish more accessible, which in turn will make people more informed and will enable them to make better decisions about invasive crayfish management. So in today's talk, we will learn about the work being done in Michigan to control invasive red swamp crayfish, specifically since their first confirmation in 2017. Dr. Brian Roth from Michigan State University and Kathleen Quebedeaux from the Michigan Department of Natural Resources will discuss the intensive efforts that they and their partners have made to control the spread of invasive red swamp crayfish. They will provide the insights into their control and monitoring strategies and then evaluate their impact. So Dr. Brian Roth is an associate professor and researcher in the Department of Fisheries and Wildlife at Michigan State University. His research focuses on predator-prey dynamics in aquatic systems and invasions of aquatic animals. He works closely with Michigan DNR to determine the best way to eradicate or control invasive crayfish. Additionally, he helps inform management actions by providing his expertise on crayfish biology, sampling methods, and supervising graduate student projects. He received his PhD from the University of Wisconsin. And then Kathleen Quebedeaux works at the Michigan Department of Natural Resources as the red swamp crayfish response biologist. Her work focuses on early detection, control, and eradication of red swamp crayfish by using an adaptive management framework. Her undergraduate degree is from the University of Georgia, where she studied fisheries and wildlife, and she received her master's degree from the University of Illinois, Urbana-Champaign, where she studied crayfish conservation, taxonomy, and ecology in the Ozarks. Thank you both so much for being here. So to everyone joining us today, let me just reopen that. We encourage you to type in any questions that you may have in the Q&A box or in the chat at any point during the presentation, and then we will go through them together at the end. And also, as a reminder, this webinar is being recorded and it may be posted on the ICC website with captions shortly afterwards. And all right, with that, we will first hear from Kathleen. So Kathleen, you can go ahead and share your screen.
All right, everyone can hear me okay. So I'm going to be presenting today with Brian on a comprehensive overview of our invasive red swamp crayfish response in the state of Michigan. I want to acknowledge all of my co-authors and especially Brennan Wright, who helped me make a lot of these slides.
Kathleen, you are not sharing right now. I don't know if you wanted to
try I'm
sharing.
not?
No.
Okay. Sorry about that, guys.
Perfect. I can see it on my end now.
Yes. Here, all of our co-authors, you can see them now. So to begin with, I want to go into just a little bit of background information about red swamp crayfish or Procamberus clarkii. I'm sure most of you are already familiar. but they're the world's most invasive species of crayfish. They're a worldwide invader, partly because they are the species most commonly found in the food industry and eaten at crawfish boils. Species also highly fecund, and a female can have over 600 eggs, and they can have multiple recruitment classes in a year, which makes them very efficient at evading new places. Additionally, once they become established outside of their native range of southeastern North America, they can be very destructive. One of the ways they're destructive is through predation on native species. They'll eat just about everything, so they'll prey on fish eggs, fish, other macroinvertebrates, macrophytes, and additionally, they can outcompete native species of crayfish. We have 8 native species of crayfish in the state of Michigan, so that is also a concern. Through those different methods, they can change the trophic structure of an ecosystem once they become established. And additionally, red swamp crayfish are known to get at really high densities once they invade a new location, partly due to the fact that they're not being preyed on and they're out competing. And they also are known for burrowing behavior. So at those high densities, the burrowing can start to cause erosion issues to banks. So that's another concern once they become introduced. Additionally, red swamp crayfish have been known to increase the turbidity of water bodies by destroying the macrophytes within a lake or pond. So moving on to Michigan's invasion in particular, in 2015, red swamp crayfish were prohibited in the state. However, that did not prevent them from being introduced. They were first found in 2017, and that was in a retention pond near a hotel in Metro Detroit. Since then, we have found them in over 30 water bodies and 8 complexes, which sounds like a fairly large invasion. However, most of these water bodies are small retention ponds, golf course ponds, or backyard goldfish or koi ponds, and they're mostly in southeastern Michigan in the Metro Detroit area, which you can see in the square on the map. Because of the fairly localized nature of the invasion, that's part of why we've had a very aggressive response, because we still hope that eradication is possible for the state. So beginning in 2017 through 2020, our main focus to try to control the population was through trapping. So we kind of ranked the ponds into different categories based on how heavily invaded the ponds or water bodies were. So the ones that received the most high intensity trapping were the ones with the highest populations of red swamp crayfish. And in those ponds, the goal was to try to achieve eradication through trapping. We placed traps every 5 meters of shoreline, and those traps were checked almost daily. The next category were ones that had slightly lower populations of red swamp crayfish, and those ponds, we had a goal of suppressing the population, so we did lower intensity trapping. The ponds had traps placed at the same density, but they were only checked about 2 to 3 times a week instead of almost daily. There are a few other ponds or water bodies in the state that are invaded. However, those ponds were trapped for surveillance purposes only because they had very small detection rates, or they were handled by collaborators in the state, such as SISMAs. So starting in 2021, we added in another tool that we started using to try to control the invasion, and that was by doing pesticide treatments. Unfortunately, there are no crayfish-specific pesticides known. However, pyrethrin and pyrethroid mixtures have been used for invasive crayfish, including red swamp crayfish in other parts of the world. So because of this, we moved forward with looking for experimental approval to use Excite-R, which is a pyrethrin-based pesticide. In order to do that, we had to receive a permit from the U.S. Environmental Protection Agency and also the Michigan Department for Environment, Great Lakes, and Energy. Excite-R is an indoor-outdoor insecticide. It's approved for agricultural, food industry, residential use, and even on pets. However, it is not labeled for aquatic use, which was part of why we needed that special permitting from the EPA. It is highly toxic to fish and invertebrates. Like I mentioned before, it's not specific to crayfish. However, one benefit of it is relatively non-toxic to mammals and birds. One other drawback with the pesticide treatments is we're not able to apply this pesticide in all water bodies at the moment. The ones we're applying it to are fairly isolated and don't have any T&E species that would be concerned about being affected by the pesticide. And we also have to receive landowner permission since most of these ponds are on private property. To date, we've done over 22 treatments at 10 water bodies, and starting in the summer of 2021, where we treated the Sheridan Pond, which is like the ground zero initial introduction site. And since then, we've increased the number of ponds and also the frequency we've treated the ponds. So in the fall of 2022, we treated 4 ponds. And then this year in 2023, we did a set of spring treatments and fall treatments. And this is to target in the spring the adults when they're mating and moving around more. And then in the fall, it's targeting juveniles because that's when we see the highest number of juveniles in the water bodies. Throughout the years, we have used 3 different methods for applying the pesticide, which our collaborators at USGS helped us with. The first year, we used a boom method of applying, and all of these are targeting spraying the pesticide underneath the water line. So with the boom, it was located on a long rope and it was pulled back and forth across the pond. It was very labor intensive and not the most efficient. So we pursued using other application methods. In 2022, we attempted using an RC boat, which is pictured here, to apply the pesticide. And that worked great until it got caught in macrophytes in the pond. These ponds have a lot of macrophytes, and so the RC boat was not able to be used in all of them. So starting 2023, we began using irrigation line to apply the pesticide, and we've had a lot more success with that. And you can see it pictured here. So the irrigation line is really nice because on the day of, it is pretty easy. You just plug it in and to a pump and start applying the pesticide. And you can get a lot done in a short amount of time. However, it does have a lot of prep time beforehand when you're setting the line, though luckily the line can stay in between treatments if multiple treatments are needed. You can see here the plumes when we're actually applying the pesticide, and we have them evenly distributed around the edges of the pond. And the pond pictured here is the Sheridan Pond, which is once again the pond where we first found red swamp in the state. Leading up to treatments, we monitor the weather forecast to make sure there's no major rain events predicted, so that pesticide is not getting washed out or downstream. Additionally, we have carbon-based filters placed at any outflows to catch any pesticide that might discharge. The majority of these ponds are fully enclosed, or there are some that have outflows, so that's where the filtration comes in. And then additionally, there is another man-made pond directly beneath the Sheridan, which is the main one where we have concern about outflow. As the treatment period goes on, we collect water samples in the ponds and also at downstream sites. And that treatment period is just based off previous years, how long we expect to see the pyrethrin stay in the water. Additionally, we use bioassays to monitor the pesticide levels, and the bioassays are essentially crayfish in cages that we place in the different treatment ponds to determine survival. As you can see at the graph here, pyrethrin concentrations decline rapidly in our treatment water bodies. In general, they're non-detectable between 4 to 14 days, though that does depend on conditions. Additionally, the mortality with the bioassay crayfish ends within 3 to 10 days post-treatment. The degradation rate varies on many things, but the main ones we think are at play here are the amount of solar radiation and temperature in the ponds. We also think that the amount of organic material and macrophytes in the ponds could be playing a role as well. Additionally, we did see non-target impacts, but they were the ones we expected. It was primarily minnows and stunted sunfish, and we saw no mammal or waterfowl impacts in the ponds. So the next thing I'm going to be going through is an overview of our trapping results and also the impacts of the pesticide treatments on our trapping in our high intensity and low intensity trapping ponds. And I'm going through this as part of showing our adaptive management plan that we have for this project, where we are constantly collecting data, analyzing it, and trying to improve our plan for the following year. So to date, we've manually removed over 170,000 red swamp crayfish from Michigan waters. And just in the high intensity trapping ponds, we've removed about 130,000 crayfish. And in the low intensity trapping ponds, we removed over 34,000. Generally, peak catches are in June and July. So I'm going to show you the trapping results for those months. And additionally, I'm going to be showing you the results only for crayfish trapped in G minnow traps that had 24-hour soak times, so that we're doing apples to apples comparisons. So there's a lot of graphs here. I'm going to walk you through them. On the left side, we have trapping results for the month of June, and on the right, we have trapping results for the month of July. And they're the same ponds. They're all high intensity trapping ponds. The y-axis shows the average daily catch per unit effort, or CPUE, which in this case represents the number of crayfish caught per trap. And you might notice also the scale is different on different graphs, but that's just for ease of showing it all in one graph here. So overall, you can see with the high intensity trapping ponds in both months, the trend is that the population is going down over time. However, I do want to point out a couple things, one of which is the time when you see the biggest dip in CPUE pretty consistently is when we had pesticide treatments, which are indicated by the red arrows here. I also want to point out that in Pear Ridge, which was not one of the ponds that received pesticide treatments, we saw a large dip in the catch per unit effort in 2022 and a rebound in 2023. And we think that might be due to low water levels that year, either artificially, or not artificially, but reducing the population or reducing our detection probability, because you can see in 2023, the catch per unit effort rebounded. Moving on to the low intensity trapping ponds, you see a different pattern emerge. So once again, on the left are the results for the month of June, and on the right are the same ponds, but in the month of July. I also want to note that the results for Fox Creek 1B were pooled with Fox Creek 1 before 2023, which is why those two graphs have less data. So overall, with these low intensity trapping ponds, we see that the populations, or at least our catch, is actually increasing a little bit over time or staying about the same. So we're not seeing that suppression that we see with the high intensity trapping. However, the exceptions are, once again, when we did pesticide treatments in these ponds, the catch per unit effort drops dramatically, almost to zero in these ponds. So it's showing really the efficiency of these pesticide treatments as compared to trapping, in particularly low intensity trapping. I want to show you one more set of graphs here. So this is the Sheridan complex represented for the month of June. And the Sheridan complex is the 4 retention ponds where we initially found red swamp crayfish that I've been talking about. The Sheridan pond is the only one of these 4 to receive pesticide treatments due to permitting reasons, and it is also the pond that is most upstream of the fort. You can see also the Sheridan is the only one that has had a drastic decrease in catch per unit effort over time till 2023, where our CPUE is almost 0. Versus in the other 3 ponds, which have not been able to receive pesticide treatment so far, catch per unit effort has been increasing or staying about the same over time. So this really shows why we want to treat the whole complex going forward, and we hope we can get permission to do that at some point because it clearly is what's needed to bring the population down. So big takeaways, first off, is that high intensity trapping is effective for suppressing the population, but it requires trapping almost daily. Low intensity trapping is not effective for suppression, and trapping is not efficient for eradication in general. It can be effective at high intensity levels, but with the amount of effort it requires, it just makes more sense to use the pesticide treatments to try to achieve eradication. Additionally, red swamp crayfish were caught after our pesticide treatments. However, it's in pretty low numbers. We think that part of the reason why we're finding them after the treatment is due to underground connections between water bodies, allowing them to recolonize the pond. And additionally, they may be hiding in burrows. As I mentioned, there are burrowing species and there are a lot of burrows near these ponds. So that may be allowing recolonization also. So all this to say is multiple pesticide treatments are likely needed to catch any straggling crayfish, and also it's important to treat the entire complex so they're not recolonizing. And part of how we're trying to be more efficient and make these pesticide treatments catch every last crayfish is by doing burrow treatments. We have been using 2 different methods to try to achieve that goal. One is with pyrethrin, which is the same pesticide Excite-R that we are using in the ponds. And in this case, we just spray it into the burrows. And it requires the same permitting process as the pond treatments. The other method we've been using that our collaborators at Auburn and USGS also helped us with is Benseal, which is a bentonite clay sealing agent. And this product is generally used for pond sealing. It's used in wells and mining. And it's actually very similar to kitty litter, where it's loose gravel. When you pour it in, you get it wet, and then it hardens and solidifies. So you can use it to permanently fill in a burrow. Both of these 2 burrow treatment types have similar mortality, so about 60%. However, there's a few things that have led us to focus on Benseal as our main burrow treatment method. One is that it helps with bank stabilization by filling in the burrows. As I mentioned, when the crayfish are at high densities, their burrowing behavior can cause erosion issues. Additionally, it restricts the burrow from being recolonized, and it can be applied throughout the season, not just during the short period when we have permission to treat with pesticides. However, it is somewhat less effective in complex burrows or burrows that are very wet at the water's edge. Excite-R is a bit cheaper than Benseal, but because of the specialized equipment and permitting, it's just not efficient enough for it to be what we focus on going forward. So we have been, especially this fall, mostly been using Benseal. Next steps are, 1, we're going to be reducing trapping, which we have already done in 2023. Trapping is mostly going to be used for surveillance and early detection. our pyrethrin-based pesticide treatments are going to be our tool that we use to try to achieve eradication. Additionally, we're continuing our bend seal treatments throughout the year, and to date, we've filled over 1,800 burrows. Another thing we need to work on is continue to evaluate the results of our trapping to hopefully continue to improve. And we're also exploring alternative control strategies, one of which is lining the banks of some of these ponds to hopefully remove burrowing habitat. We're modeling that project after project conducted by Wisconsin DNR. And we're also hoping to deepen some of these ponds with dredging, and that way we can hopefully implement some biocontrol with larger fish such as largemouth bass. And then one other project that's still at the beginning stages is we're hoping to do some neo-female work where we stock neo-female crayfish. And we're only at the very early stages of that. So we're going to hopefully start modeling the populations using our trapping data that I presented here today. So with that, it's Brian's turn.
Thanks, Kathleen. I will start sharing my screen if I'm able to. There we go. I think it should be showing up now. Okay, good. So at MSU, we work hand in hand with the Michigan DNR to try to accomplish these management objectives. But along the way, we're not only interested in how those treatments work, but we're also interested in a suite of questions that we hope will aid in our ability to control their populations. And so there's a few other questions here that we've needed to answer, but I'm going to talk about some of the kind of other projects that MSU has been involved with throughout the removal period. So the first one we hope to address is, where did these crayfish come from? Certainly, we had some ideas, and here I have native in quotes. That's because while certainly red swamp crayfish are native to Louisiana, in Alabama, it's sort of a debate, and it depends on maybe where you are. So there's some debate about that, but certainly it is a potential source. We also had genetics records of crayfish captured from other invasions, including the one in Wisconsin that Kathleen mentioned. Ruben gave us some crayfish from Chicago. And then we also tested two other sources, including 2 orders of red swamp from Carolina Biological, the biological supply company, as well as the seeder from Grand Rapids at a live food market. This work was led by Kim Scribner's lab and Nick Sard, who published a paper on it. And basically, what I want you to pay attention to here are the different colors. And so if you see all this green here, that indicates higher relatedness. Different colors and these skinny bars represent genotypes of individuals. And so, for instance, those crayfish from the Grand Rapids seizure here are all pretty, are very much similar, right? Whereas those from Carolina Biological here, there's some diversity there. And so what this indicates is that in this FAST structure is that this is, the top one is where the marginal likelihood is maximized, which indicates 7 different groups. And the bottom is where nearly all ancestry is captured, and that's 8 groups. But one of the things that I want to point out here is that in the Michigan invasion, the colors don't match the potential sources. And so what this indicated to us is that the invasions in Michigan are probably from multiple unknown sources. Certainly those individuals from the biological supply demonstrate relatively high diversity, and you see a little orange there, but nonetheless, we're not exactly sure where they came from. Certainly, what this also indicates is there's some evidence for founding at Sheridan Pond and spread to Fox Creek. We wanted to explore this even further, and this is a similar type of plot, but it's based on discriminant analysis of principal clusters. And what this finds is that if you look at the x-axis here, these are all the different ponds that have been invaded. And I'm not going to go through each one, but you can see that they group relatively tightly here. So what this indicates is 8 groups of highly related crayfish within a single group. So for instance, the Sheridan complex that Kathleen mentioned is, where is it now? I lost it. Oh, here in dark blue, sorry. So in dark blue, this is the Sheridan Pond, and you can see that they're all very similarly related. Each of these little white lines is just a split amongst the ponds. So not surprisingly, all of those individuals among those 4 ponds are pretty tightly related. When we parse that, all those different invasions, what we see is kind of 3 main groups. So this River Oaks groups here is off on an island by itself. A relatively new epicenter of crayfish invasion in Shelby Township is also kind of by itself, indicating pretty distinct genetic grouping. However, everyone else is over here in this right-hand corner, which indicates that they're relatively closely related, at least compared to River Oaks and Shelby Township. When we look at these a little bit closer, what we see is that here's that River Oaks complex. And so, yes, there's some overlap in their relatedness. But when we look here's that dark blue of the Sheridan complex. These guys are all very, very closely related. This is a nearby golf course that comes out as a light blue color. And this is a separate kind of group of ponds. And so what we see is that they are indeed separate, even though they're relatively closely grouped on that previous plot. One thing that really strikes us is this, one of the suite of ponds here on a single golf course. And what we see is actually 2 groups of crayfish on a single golf course. So here we have one group of ponds on the right-hand side and another group of ponds on the left-hand side. So even though this is on a single golf course, there is some genetic differentiation between those 2 populations. What we think is that access to Fox Creek actually may act as a barrier to gene flow among these ponds. And when we look graphically here, I'll just direct your attention to the lower right. You can see that golf course, and that these upper right ponds are of one group, and the ones on the left-hand side are on another group. Now the creek kind of runs relatively close to the midpoint of the golf course, but certainly it seems as though the creek has a certain role that we're still exactly trying to figure out, but we think it may be a barrier. Yeah. Another project that we have been working on is understanding crayfish movement. And certainly one of the points here was to try to support some of those genetic analyses to help identify populations that may be acting as sources. So those crayfish that are leaving more commonly on some ponds than others, but also as a means to try to make trapping more efficient. So we could then identify habitat preferences. And also, because one of the techniques that we're using is radio telemetry, we can actually use this to inform something about life history, including burrowing, because we can track crayfish into their burrows. And then lastly, we implemented an acoustic telemetry experiment down here on the lower right-hand side to determine whether and how crayfish survive treatment. Sorry, it's not survive a treat, it's survive treatment. The last 4 letters got cut off, sorry. So for this talk, or this portion of the talk, what I'll do is compare radio and acoustic telemetry for monitoring crayfish movement. And some of the metrics that we'll be using, including distance, move, and home range. And our hypotheses are interesting because really we don't know a ton about crayfish movement. And so really we'll find something worth noting. And that's just because crayfish movement studies are really rare, and in lentic systems are even rare. So I could find 2 publications on crayfish movement in lentic systems. And for acoustic telemetry, this is relatively novel. I found 1 thesis, and I know Jim Stuckel's lab at Auburn has done this a number of times as well, but still waiting on perhaps publications from that. So just to give you an idea of the effort that we put into our telemetry, I'll just kind of skim over the models and stuff like that, but this occurred right around that first treatment of Sheridan Pond Complex, and we tagged 34 crayfish with radio tags and 36 with acoustic tags. And if you look here, Aaron is a real hard worker. However, it's difficult to match the 2-second pulse interval associated with the acoustic tags with radio telemetry. In addition, we're able to get much higher resolution on acoustic tags than we are with radio, but we can track over a much longer time period. So for radio, we got 355 detections, which is excellent for radio, given the number of tags and the number of ponds we were monitoring at the time, in addition to Sheridan. But for acoustic, we had over 12 million detections, giving us a really fine scale view of crayfish movement. And so when we look at this data, what we see is some crayfish moved as expected. So this is a colored plot of crayfish movement, where the earlier times are here in blue, and then they move up to red as later. So it moved around some and then died on treatment day, and it just didn't really move thereafter. This crayfish had a relatively small home range and less than 30 meters a day of movement. And then we saw some behaviors that were completely different. And so this is a different individual. It was tagged around the same time, died on the same day, and its home range was the entire pond, and with movement rates exceeding 100 meters a day. And just to give you an idea of what this looks like when we move it through time, you can see that this crayfish is really almost just doing laps around the pond, which was kind of surprising to us. I don't know if we'd seen that behavior before, but certainly it indicates higher movement. When we look at the home ranges here, plotting with 95% minimum convex polygons, so with radio telemetry, they're a little bit more jagged, but you can see that there's some differences here. With the acoustic, I just showed a subset of the tags because otherwise it would just look like a big blob of, I don't know, color. Just to give you an idea of the type of resolution that we see. Each of the dots there represents a single detection. When we compare those home ranges, what we find is that with acoustic, we're really getting a bigger idea, or excuse me, a bigger estimate of home range size. And so with acoustic, it was about 2 to 3 times as large as radio, although the variation, according to these box plots, is fairly similar. One thing to note is that we did not observe onshore burrowing with tagged crayfish in Sheridan Pond, which was somewhat unexpected. Work by Maggie Ravelin has really refined this work during her time at USGS. And what she found is that those crayfish that were demonstrating those big loops around the entire pond were all reproductive males. And in fact, reproductive males showed drastically higher home range than either form 2 males or females. And those two crayfish, male form 2 and females, have relatively similar home range size and behave relatively similar, whereas those reproductive males are doing something completely different. Maggie also did some analysis of behavioral states and breaking this down into different phases by activity. And what she found is that these crayfish alternate between encamped behavior, where they're moving relatively small distances, as you can see in these kind of orange dots, and then exploratory, where they'll go out and move much faster over a short period of time. When we compare again those three different reproductive forms, females, reproductive males, and non-reproductive males, we can see that the state 1, which is the encamped state, is much higher for females and form 2 males as opposed to male 1, where many of them are really moving around the pond substantially. Another project that Kathleen indicated was the evaluation of fish predators as biological control. And we really wanted to determine whether fish could act as biological control agents, simply because we can't just treat every single pond with chemicals just based on because they're private or for other reasons. And so we wanted to evaluate the predatory capacity, particularly of native predatory fishes. And one of the ways to do this is to develop optimal foraging curves for these various species. This work was led by my graduate student, Samantha Strandmark, who just defended her master's in July. And so a lot of this work is to her credit. So really, we asked a few questions. What size of crayfish might we expect predators to consume in nature? And what we expect this to do is represent a nadir in a cost-benefit curve, where the benefit is the denominator of that ratio. We also want to know if predators differ in size-specific consumption rate, and we'll just look at, based on crayfish size, how the consumption rates differ across species using logistic regression. And then lastly, we wanted to look at crayfish sex and how that affects handling time and consumption rate. So we ran a pretty substantial experiment, or Sam did, and we looked at 3 different species: largemouth bass, green sunfish, and bluegill. We chose these species for a couple different reasons. One is that they're native to the area. 2, we know that they can survive in the ponds that the crayfish are already in. And 3, they're relatively easy to obtain if we desire to stock. We ran a bunch of different crayfish sizes from 4 millimeters carapace length all the way up to 51. And then we grouped them into bins for analysis. So in all, Sam fed 459 crayfish to these different predators. And when we look at log handling time here on the y-axis versus carapace length, not surprisingly, largemouth bass handling time is lower than the other species, whereas bluegill increases relatively steeply. And if you know green sunfish, their morphology is relatively intermediate between those 2 species, and their handling time is similarly intermediate. When we look at those cost-benefit curves here, so we estimated cost as handling plus pursuit time of those predators, and the benefit simply as crayfish mass here. So that's how much mass that they're consuming. What we saw is that the nadir for bluegill was at 14 millimeters carapace length. So this represents the optimal size of bluegill consuming red swamp crayfish. And for green sunfish and largemouth bass, it was similar, whereas largemouth bass had a pretty large basin where crayfish were relatively optimal. When we looked at the probability of consumption, we see the results of handling kind represented here. So at small lengths, largemouth bass and bluegill had the highest probability of consumption, as represented here on the y-axis. And largemouth bass had a really broad area where the proportion of crayfish consumed remains over 50%, well beyond 40 millimeters carapace length, only dropping to less than that thereafter. When we look at reproductive forms, what we see is of interest probably, and add some inference to Maggie's identification of encamped male form 2 crayfish. So what we see is that largemouth bass only, represents only largemouth bass consumption, ate every single male form 2 crayfish that was presented to them. And only those male form 1 crayfish declined relatively rapidly. Lastly, let me check the time here. Okay, still okay. Lastly, I'll talk about how our intensive management has affected native crayfishes. And as Kathleen indicated, we trapped at 2 levels, one relatively intensely and one less intensely. When we look at those trends in catches in high intensity ponds, here we look at the percent of catch versus natives here in dark, and the percent red swamp of that catch in white. The overall catch per unit effort, as Kathleen indicated, tends to go down in most of these ponds, whereas the proportion of native crayfishes tends to increase through time. This is most apparent in Meadowbrook 1, where the proportion of native crayfishes exceeded 80% in a couple years. On Pear Ridge and Sheridan, we didn't see any native crayfishes pretty much ever, with the exception of Sheridan Pond, where in 2022 we observed our first native crayfish that we had ever seen in there. So even though we're not exactly sure how they're getting in there, they did. We see a contrast in that by looking at those moderate to low density trapping ponds. What we see is that CPUE tends to either be stable or increase in some of these ponds. And only in Fox Creek 1 did we see an increase in native species. In all the other ones, particularly here demonstrated by Fox Creek 17, we see that the proportion of natives tends to decrease. Oops. There we go. So when we look at the average daily catch per unit for native crayfish only, here's the high intensity ponds on the upper panel of graphs and the moderate to low intensity trapping on the y-axis. Prior to treatment, what we saw is that the catch per unit effort of native crayfishes in all 3 of these ponds either increases drastically or remains relatively stable, perhaps with the exception of Fox Creek 16, but certainly they're holding on. Whereas in those moderate intensity ponds, we can see that the daily CPUE is uniformly going down. In Meadowbrook 1, that was one of those ponds that we could not treat or did not have permission to treat. And we can see that, in fact, the native CPUE is continuing to remain relatively high. Whereas in those other ponds, we all treated, and not surprisingly, native crayfishes decline, although they still are present, at least in Fox Creek 16B. So clearly, these red swamp crayfish are an ongoing threat, as represented by this 2,000-pound bust of crayfish on the Port Huron-Sarnia border. In addition, we find about 1 new epicenter of crayfish a year that's unrelated to those populations, or at least we think they're unrelated to those populations that we're already aware of. My master's student, Rachel Fegley, is looking at crayfish invasion pathways, and she executed a survey of aquarium stores around the Great Lakes. and this spans all the way from Illinois, and with partners, we actually have some data from Wisconsin and Minnesota as well. But you can see here that she found a total of 8 different non-native species across the Great Lakes, including marbled crayfish and this beautiful Australian species of crayfish. Yes, it was that big. It was gigantic. And so, although this is a different figure than what Kathleen put up, certainly we're following those guidelines from adaptive management. We are continuing to learn about how to control crayfish through experimentation. The ecology and biology of these creatures obviously is critical, not only to make our actions more relevant, right? So in this case, we want to make sure that we have an idea of what their impact is on the ecosystem, but also to provide evidence of rehabilitation recovery after these treatments. If we can observe how these ponds respond in a positive manner after we treat, then that adds more fuel to the fire to continue intensively managing the species. In addition, we gather information about where uncertainty lies. And so one of those uncertainties is certainly has to do with their impact in these Michigan ponds, for which are pretty understudied. And then we adapt, right? So one of the next things one of our highlights of this program that we've done is rapidly take experimental results from colleagues such as Jim Stuckel and Annie Allert, as well as others at USGS, and implement those in the field. And that sort of rapid adaptation from experiments to field is one of the highlights of our program here in Michigan. One of the things that we're doing is executing a structured decision-making exercise here. In fact, Lucas, if you're on the call, I filled out the poll to get that appointment done in order to strategize our next steps. And so we really are trying to learn from what we've done already and adapt it towards a more effective management process. And so with that, I'll end. Certainly in terms of thank you, there's a lot of that trapping data that you saw was executed not only by the DNR, but also a whole bunch of undergraduates here at MSU. The Great Lakes Restoration Initiative has funded our project over the last several years, of which we wouldn't be able to do the work that we're able to do without it. The Michigan DNR, there are several folks that assist us regularly on this project and have endured some pretty harsh conditions over the years. So we thank you, those colleagues at Auburn University, including Jim Stuckel, those folks at USGS and Michigan Eagle, including Darren McCullough, who has approved our permits. So thank you, and that's where I'll end.
Great. Thank you so much, Brian. And thank you also to Kathleen for sharing the fascinating research and work that you guys are working on in Michigan. So right now we're going to open it up to questions in the chat or in the Q&A. So if anyone has any, feel free to type those in and we will go over them. But while we're waiting for those questions to come in, I did have one for you too. So from the movement trials, you mentioned that you see that the reproductive males were more exploratory, and I'm wondering if you guys see that the pesticide treatments are affecting these types of crayfish compared to those that are less exploratory, and so if that relates to the efficacy of the chemical treatment.
Yeah, that's a really good question, and one that I wish I could answer, to be honest with you. Certainly, Maggie's analysis is relatively new, and so we're not exactly sure. One of the things that we hope to be able to do, and I've looked kind of briefly at the data relative to their behavior right around the treatment period. And I didn't see anything that indicated one type of reproductive form or sex was more susceptible to the treatment than the other. But I think that we need to do a little bit deeper dive into the data to be able to determine that for sure.
Thank you. Currently, no questions in the chat or in the Q&A. But again, I have another one. I have a whole list if we had time to go through them all. But recently, from my experience, I've seen that we've seen a really big uptick in non-native crayfish conformations around the area, like in Minnesota with the signal crayfish, and in Canada with the marbled crayfish. I'm just wondering if you guys are collaborating with these, since they're pretty close to Michigan, if you're collaborating with these state agencies and trying to manage these invasive species in the area.
We have spoken with folks from Ontario some. The signal crayfish finding is a little more recent, so we haven't gotten to talk to them yet, but we were able to share with them some of the permitting information we had, because I think they are interested in potentially exploring that. So yeah, we'll hopefully be able to stay in touch with them as that progresses.
Yeah, certainly we're open to it. And so if we can offer something to folks in those areas, that anything, we're always happy to talk about it or help out wherever needed.
Yeah, that would be great, just because it's an ongoing threat. Pretty scary. Another question is, how are you both engaging with the public regarding the presence of red swamp crayfish in Michigan? Are there any educational or outreach programs in place to inform local communities about the efforts being made and the potential impacts?
We do a lot of different sorts of outreach events. So I know I've personally been at some events at the Outdoor Adventure Center in Detroit, talking to the public about invasive species. We have printouts of what red swamp crayfish look like. One of the biggest hurdles I think most people here probably are familiar with is that many people just aren't familiar with crayfish, and that there's more than one species, much less that some are native and some aren't. So before my time here, our team developed a field guide to help people identify the native and invasive species, and that's available for free online. And we're also hoping to create some more like 3D models for educational purposes of different species in the state. I'm sure Brian has his own things too that he's been involved with.
Yeah. So every year we do a couple different outreach events at camps and schools. And so a few years ago in particular, I had a technician that would wear a crayfish costume into the school. I don't know what happened to that crayfish costume. I wish that he would have left it at MSU, but it's not there. And in addition, as part of Rachel's work, we received an interjurisdictional grant, and we're working with MSU Extension as well as others from around the basin. In fact, a lot of people that are on this conference call, and we're developing an outreach poster because, in particular, for ornamental crayfish, the crayfish guide, which is great for those crayfish that are in the wild, the colors that you see in the aquarium trade are wild, right? And so we're developing a poster that hopefully several of you will see in the relatively near future, to be handed out at aquarium stores and other sorts of water gardening or other sorts of trade shows as well. So yeah, we're trying. One of the things that we find really important is that outreach piece, and so we're kind of ramping that piece up.
Yeah, and I'm glad you pointed that out, Brian, because one of our most recent sites where we found red swamp in the state, we actually found a strange color morph of like a mostly white red swamp that usually you see in the pet trade. I had never seen in the wild before, and we found multiple individuals with that color pattern. So I think that's going to be really useful. And another thing that came to mind is we know that one of the biggest sources for new introductions for red swamp crayfish are actually classroom pet releases. So I've been working with some other folks in the DNR to try to get information out to teachers about the importance of not releasing any aquarium pets, and also that it's illegal to have live red swamp crayfish in the state.
Great, thank you so much. And I know that we're a little over time, but we have two more questions from the chat that I think are really important to address. So one question says, you found that some crayfish demographics had more movement than others. Do you think that even those traveling crayfish return to a given site eventually, or do they leave home and stay away?
They die in a treatment, Ruben. We're not exactly sure. The radio telemetry gives us a slightly longer time window in which to evaluate movement. And yeah, I mean, these things, I would love to work on it with someone, because it kind of defies easy categorization. So certainly some crayfish had pretty defined home ranges over weeks and weeks, if not months, and some just were all over the place, even jumping from a pond out into a different area. We saw that a couple times. And some that were just homebodies, some that mostly spent their time in a burrow but would come out occasionally and then go back in the burrow. It's really hard, given if we could trap every crayfish or tag every crayfish and observe them, I'm sure that we could have a little bit more inference from the data, but that's impossible. So at some point we're going to really work on quantifying that, especially the radio telemetry data that's over a longer time period. And if you have any ideas, let's chat.
Great, thank you. Last question. Do you expect your findings to translate pretty well to larger water bodies, both in terms of the efficacy of pesticides and the effectiveness of other management actions, or is that still to be determined?
I think a lot of that is still to be determined. As I mentioned, we have a lot of permitting things that are difficult to navigate, and bigger water bodies are definitely something that adds that. We only have one larger water body that we know of in the state that is infested with red swamp, and we've had not as much success trapping them there either, and we don't know if there is genuinely a crash in the population there, or if it's just a detection probability issue. And that's something that we hope to try to look into more in the future. And we work hand in hand with the SISMA that's located there, as well as Gun Lake Tribe. So that's definitely something that's been on my mind. But yeah, I think that it's definitely a very different system, and the things we've learned could be helpful, but are definitely more targeted towards smaller water bodies.
Yeah, I mean, those larger natural water bodies are difficult to handle, because certainly you can't just dump a bunch of Excite-R in those large natural lakes. So, that's where the biological control aspect of the project also becomes relevant, in that if there's a possibility of changing fisheries regulations that could increase red swamp crayfish mortality, we should explore that, right?
Great, thank you guys so much. That's all the time that we have today. If anyone else has any other questions, you can email me your questions, and then I'll make sure to forward it over to both Kathleen and Brian. So thank you again so much, Kathleen and Brian, for coming and presenting your work, and presenting the work that you and your other collaborators are working on. And thank you to the attendees for joining us today. I hope everyone has a wonderful rest of their days. Thank you.